Fernanda, for the introduction. And also thank you, NODCC and IRC5 and you all for allowing me to be here. Thank you so much. So I'm going to talk a little bit about what can MRI tell us about a person with DCC or CCD, as also Professor Linda Richard said this morning. Also, when I mean DCC or CCD, it's the same thing, so disorders of the corpus callosum or corpus callosum disorders in a couple of slides. So, we already saw, I think now we're going to see the slides. Yeah, so we already saw a lot of brain images today, and I think that Professor Gregor Kasprin made my job here much easier because he explained us all about the diagnostic images, about what we should look for when we're doing clinical MRI. I will try to talk a little bit about a different way to look to the brains, and not only look into the anatomical details like in the clinical MRI, but also show what these tools, what we can also understand, and use that to understand not only about all the connections in the brain of a person with callosogenesis, but actually how these tools can make us to understand how the normal brain or typical brain and also the non-typical brain functions and actually reorganize and is reorganized during development and also the whole life. What I'm trying to tell is about the concept of brain plasticity. We know that our brain and our circuitry is always changing, and somebody in the audience today already mentioned that what we already know well also is that our brain is much more plastic in the beginning of our development than later, but there are a lot of evidence that we already know that it can, yeah? Not yet? Almost? No, so I was just to say that we always were interested in understand how the brain functions. And during a lot of time, actually, we actually could also only have access after, like in necropsis, or when we, for example, a person had a problem with the language, and then we only could know afterwards. But we know that with MRI nowadays, you can have actually pretty detailed images that we're already showing today. And not only about the anatomy, that really is very similar, or we can match a lot of what we can do, and we still have to learn a lot about with histology, but also we can have details that are also related to the brain function and also different structures in the brain. Those images that are very high resolution images, they also allow us to reconstruct the images like in 3D dimension, as we can see here, for example, with the reconstruction of the gyres and the verifications of the brain. All those techniques can be combined and joined. So here we can also have just an imaging of activation of the visual cortex. So inside the MRI, you can actually stimulate the vision, or you can ask the person to really just move the head, the hands, and you can map where those areas are functioning during the examination. Those are called functional MRI. And here we have an example of an stimulation of the visual cortex. Here we are seeing the back of the brain. Here is the front of the brain. And here, for example, we have a slice like that. And we are seeing here an area that's responsible for the movement of our hands. So we compared our brains when we are doing nothing and when we are doing something. Okay? Those are called functional MRI. And the mapping of those, we know that the brain, the function is much more complex. And they link actually, usually not only one region is responsible for the functioning or the behavior. We need multiple connections between different areas of our brain to really have this functioning complete. And actually it's probably because we also need a very robust structure that connects these different points of our cortical. The cortex is the periphery, and it's needs to be connected in many ways to really produce this vast repertoire of function. But with those techniques nowadays, we can actually map those circuitries and actually reconstruct what we call would be a typical brain connectivity, in meaning functioning and also meaning in terms of structure. And understand this brain connectivity, how it functions, and also how it's connected, it helps us to understand what we expect from a typical brain and also what we expect in a non-typical brain. Comparing these two, like reconstructions of our connections, we can learn a lot about different types of pathologies, understand how the physiopathology of those disorders, and so on. So here's just an example of reconstructions of those connections made by this typical examination of the MRI. So it's called diffusion MRI, and with a post-processing, you can actually reconstruct the tracts as we already saw in the previous meetings. So just here, the corpus callosum is this red here. That's because it's color-coded in red because it goes lateral to lateral. But we have all the other tracts here also reconstructed and go in different directions. Again, combine the functional MRI with the structural MRI, we can map our expected brain connectivity in the typical humans. And we can also understand what's going on in terms of reorganization or lack of structure in different pathologies or also in non-typical individuals. We have to keep in mind, however, that everything that we are seeing with MRI is in a micro scale. We will never replace what we can really see in terms of the real connection of the neuron, the final synapses. We will always need to complement or to interpret those techniques with the limitation that we are seeing in a micro scale, and not in a macro scale. I'm sorry. Sometimes because of non-native speaker, we for micro scale and macro scale is difficult. Anyway, you understood. Yeah? Okay, so just here, giving examples that we can map that in different conditions, like in autism, in callosidogenesis, in Zika, and the main purpose of that is actually to understand the pathophysiology of those brains, and also maybe to be able to guide better diagnosis, to guide better therapeutics, to understand better all of those. And however, what we are calling a typical connectivity, we also need to keep in mind that it's changing over time. It's changing in normal brains. We actually, to be able to form our typical or expected connections, we need our genes, and we just saw a lot about our genes and how important it is, but also experience is very important to shape our connectivity. So, for example, a kid, when it's developing, when the synapse is related to the language, is when it's exposed to the language. We all also know that in many conditions, actually the brain is shaped differently because they didn't have access to that experience. It happens, for example, in blind people. They never saw. So their brains, they form the connections in a different way than the ones that always had the vision in the place. And it also happens with different conditions. When your brain is actually faces during development something that's not expected, like the lack of the presence of the corpus callosum, it is also reorganized in another way. And that's what we are trying to understand in our research, and together with Diversify. It's a kind of the shape of the connectivity related also to brain plasticity. And we know that brain plasticity is much more robust in the younger or in the immature brain, but we know also that it happens through life. Yeah, and we need, and we always need to understand that we can always challenge our brains because we are in constant changing of that. Actually, in the normal development of the brain, the signatures also change. See here? So this is expected what's happening in a newborn. We have a lot of secrets that goes, other retracts. So these changes are all expected. And those changes can also be mapped by different techniques, including MRI. Those are just some examples. I'm going fast also because of the lack of time. And here we have examples of prenatal images and postnatal images, and see how the images they change, and those are expected changes. And here we also can see this diffusion image that we have explained that are also changing on time. The clarification is always changing on time. And we can use MRI to understand that if something is not going as expected, then it calls attention. And this can also be related to a biomarker of not normal development, for example. Just another example here, one of the biggest structures that we always look is the corpus callosum. So here the corpus callosum in an ultrasound, and here is the corpus callosum in a fetal MRI. But as we already saw, the corpus callosum during development and in DCC or in callosogenesis, you can have a complete callosogenesis, nothing. Here we don't have any piece of the corpus callosum, or we have the partial, we have a hypoplasia, and we have here a lipoma. Who was here before in the morning? We had actually an excellent question related to lipoma. So something that's there in the midline that also didn't allow the neurons that were expected to cross to be able to cross. And we have other conditions that also change the corpus callosum, like infectious disorders. Just here in Brazil, you might know that we had the epidemic of Zika virus, and looking to the MRIs, the prenatal phase, we could understand that something was not going on. And all the changes could be mapped, not only related to microcephaly, that was a more actually announced and were more in the media, but actually those kids had a lot of change in different aspects of the brain development, including the corpus callosum. So for example here, we have a thinner corpus callosum in one of the kids that were exposed to Zika virus, and also other changes in the brain, as we can see here. But also we had some cohorts, a couple of kids that they didn't show a lot of microcephaly, although they were exposed to Zika. What we don't know is that actually during the development now, that the kids are around 5-year-olds, if they show something that is not expected in terms of brain maturation. And we can use, for example, measurements in the corpus callosum to understand if something is going not as expected with this brain maturation of the kids. So here is one of the things that we can do with MRI. Again, a lot of the slices here of the sagittal view of our brain, and we can measure the corpus callosum in different ways, and then we have the measurement that's expected for certain age. So 6 months, 1 year, 1 year and a half, and when we look into the expected measurements that were expected for the Zika kids, it seems that something is not going so well. So it might be that the maturation of the brain has some effects that were caused by the Zika virus, but we are still exploring that. So this is a way to look to the change in terms and use actually the measurements of the normal, the expected measurements of the corpus callosum or different structures to understand that something is not going as expected with the brain developing or the brain maturation. Again, here we have an example of expected development of a typical brain. In the same age, we have here a DCC, a complete DCC, and here a partial DCC. But when we also, Dr. Elliott just showed that how much is important to not only look to the change in the corpus callosum in DCC. We have to look to the other changes that can be present in the brain, because this can be related to the prognosis. And again, we can have different chains together associated with a brain with DCC that could be related to the formation or not of the gyrification, like you can see here. So here is one that we have like a bacteria, so we have not very well folded brain, here non-folded at all, and different changes that were already related to genesis of the corpus callosum. And this is a paper from Dr. Scher's group and Orit Glenn, that she points out the fact that we always need to look to the rest of the connectivity, not only to the corpus callosum connectivity. And to look to these different chains in the brain, not only to the chains in the corpus callosum, is very important for prognosis. But it's also, I want to call attention now for another way to be able to look for different things in the brain without the corpus callosum. I was talking before about plasticity. And in callosogenesis, the brain is challenged because it is formed with the lack of the possibility to form the major white matter bundle. What happens with the rest of the connectivity and what can be going on with the brain with a person with callosogenesis? Dr. Richards today also pointed out one very important work in the neuroscience field that actually gave to Sperry the Nobel Prize in 1983. So he described the brains of people that has the corpus callosum cut because of epilepsy and showed that behavioral is a lot different, very different from the people that is born without the corpus callosum, as we can see here in this imaging. And actually, when we look to the brain and see the connectivity of a person without the corpus callosum, we already see, and was already showed here also this morning, changes in the white matter organization. That actually is a rewiring, is a reorganization of the connectivity or the structural connectivity of a brain with a person without the corpus callosum. So here we see a sagittal view again of a person with the whole corpus callosum, so a typical person. And here in red, the picture of the corpus callosum in that sequence that's color-coded. And here we have three different subjects. One with total agenesis or complete agenesis of the corpus callosum, hypoplasia over the corpus callosum, and here remnants, just partial corpus callosum with the remnant, the geno as a remnant. Let's take this example here. We see two stripes here in green. Did you see? So here we see the progress bundle in each of the hemisphere. Those are, as was already explained today, neurons that couldn't cross the midline, and they form these two different bundles in each one of the hemispheres. This is already a robust sign of brain plasticity or reorganization. And here what we can see are the reconstruction of the progress bundle in red, using the technique that's called tractography. And in green here, we have another abnormal bundle that's also seen only in people, or in the majority of the people that doesn't have the corpus callosum, but they have the geno, or they have partial corpus callosum. And this bundle is called sigmoid bundle, as you can see here, reconstructed in green. And it was also showed here in the T1 or conventional image by Dr. Lin Po in this nice review. So those are already signs of structural brain plasticity in callosogenesis. What does those bundles do? We don't know a lot. We need more research to be able to understand. But we do have a lot of evidence combining different techniques, so combining MRI with, for example, electroencephalographic studies, where we see the signs of the neurons. Yeah, we have different evidence that they are functioning and they are there. But what is the impact on behavior? We don't know. We need more data with a larger cohort of people to be able really to understand the impact of all these changes in the brain in the behavior. And that's why we have IRC5, for example. But those two bundles that I explained here, the sigmoid bundle and the Probus bundle, although they are signs of brain plasticity, they don't really explain the fact that we have communication between the two hemispheres of a person without the corpus callosum, yeah? And we have a lot of evidence that it happens. That's the major difference between the people that is born without the corpus callosum than the people that have the corpus callosum cut, or we call it the split brains. So, and also behaviorally, so this is one way to look to that. So you can have transferring of information from one side to the brain to the other side to the brain when we have degenesis of the corpus callosum. It doesn't happen when you have, for example, a split brain. And what explains that the two hemispheres is actually connected can also be measured by MRI. And here we have maps of dysfunctional MRI showing how the two hemispheres are talking to each other, for example. So just take here, in each of one of those squares, we have the map of a person, a typical one, and a group of people with callosal degenesis. See how much similar are the activation? Yeah, here too, very much similar. So those two regions, they are communicating to each other. Here, for example, in a person that has the corpus callosum and a person that doesn't have the corpus callosum. How does it happens? We don't know completely yet, but we are looking for those type of information. So this is another way to see almost the same image that we saw before in a different course. This was actually done in Rio, where before it was done with Dr. Elliott and Dr. Limpo. What we see here in red and in blue are the two regions that are talking to each other in a person with callosal degenesis. And here, in that side here, a person with a complete formation of the corpus callosum. So those two regions, in red is on one side of our brain, and in blue is the other side of our brain. They are talking to each other, measured it by functional MRI. What you're doing, the link between these two regions in the typical people that has the corpus callosum, it's done by a bundle, a part of the corpus callosum that can connect structure to one side to the other side of the brain. What happens with the people without the corpus callosum? We are still investigating, but it seems to be in some of cases, in a couple of cases at least, connections through different parts of our brain that cross the midline. In this case here, for example, was crossing through near the posterior commissure. So in a different way, this might be one of the ways that our brain is compensating and creating pathways that can communicate one hemisphere to the other. This is still a lot of work to be done. Not only by one group or the other, but again, this information has to be interpreted together. This is another cohort also, Vanessa Sefredi, that was here, I think that's in the other meeting. Yeah, she found, for example, those abnormal bundles in 30% of her case. But again, a lot has to be done. And everything that we are seeing with MRI, why it's so important to link clinical with translational research. Everything that we see with MRI is a reconstruction and has limitations. Since the beginning I was talking about limitations, and we have a lot of limitations with those type of results. And to be able to really understand the real connectivity, we should go to a micro level, or be able to understand if it's also happening, for example, in animal models. And this was done by the group of Professor Linda Richards, and also with Diego, that's here in the audience. Diego was then studying a type of mice that develops the genesis of the corpus callosum naturally in a good amount of subjects now in the mice. And what he saw was that actually in this mice called bulb C, he could also see when they did have the callosogenesis, he also saw probes bundles that are reconstructed here by a MRI that was done in the mice. Very similar sequence that we done with humans. And here we can see that we have the probes bundles in humans and the probes bundles in mice. And again, he also found the sigmoid bundle in the mice that was very similar to also the shape of the sigmoid bundle in a person with callosogenesis. But what's very nice and what's the coolest part is that then you can do and look to the histology. Then you can actually confirm that the sigmoid bundle that we saw by an MRI in a mice was actually there also with real connectivity tracing with histology. So again, this is all translational and a lot of things to be understanding, not only in the mice perspective, but also in the human perspective. But those are puzzles that are going to putting together to be able to understand how actually is the connectivity in our brain, in typicals and atypical people. Another way to look to that is things like that, to see maps of this huge connectivity. And this is another way to interpret those type of sequences, and we can compare a group of patients with callosogenesis with a group of people with partial callosogenesis or total callosogenesis with the controls. And look into the maps that are constructed or reconstructed in those populations, we can guess a couple of informations. For example, again, I already said that we have a lot of the connectivity, the functional connectivity in people with callosogenesis is very similar to the functional connectivity in people with the corpus callosum. And in a different way, in a different approach, different from the ones that Dr. Lin showed, that I showed in the paper, Diego now just compare the maps that are put out in terms of functional MRI from people without the corpus callosum, total callosogenesis, and compare it to typical ones, and the difference between those two maps is that here, nothing. We have actually very similar functional connectivity among those two populations, people with corpus callosum and people without the corpus callosum. When we look to the structural level, however, there are a lot of things that are going on. So a lot of reorganizations of these white matters that probably compensate in different ways. We don't expect that it's just one explanation for that, but actually this reorganization compensates and actually produces a functional activity that is there and is talking to the two hemispheres. So just to finish now, do I have Jeff is helping me with the time. I just want to show all the types of researches that we can do when we have the consortium. We already had many talks previously. They pointed out that folding is something that seems to be affected when we don't have the complete formation of the corpus callosum. Our brain is actually folded in a very specific way, and it seems to be conserved. This way of folding seems to be very preserved during evolution as well. And one of the theories that actually explains or can explain the folding of our brain is related to the tension that the structural connectivity makes our brain actually folds as it is. You can imagine that it's probably, if you don't have your major contour linking the two hemispheres, the folding might be different in people without the corpus callosum when compared to controls. Again, this is a type of question that can help us to understand the folding in callosogenesis, but can actually bring to us a lot of information about the folding in everyone's brain. So we are trying to put, for example, to be able to ask those questions, we need to map like how the brain is folding. It's also done by MRI. we can reconstruct that into 3D view, and then we have a bunch of measurements that will show how the brain is folding. And actually, I will not get into details. What we have seen so far with preliminary data is that actually the brain seems to be folded pretty much similar in callosogenesis when compared to typical people. However, the balance between the two hemispheres might be a little bit different. So actually, we need the corpus callosum to be able to actually produce the chains in the left and the right hemisphere. And without that, we might have something that is not so it's not so expected as in people with the corpus callosum. But what I really, this is still a research that's going on, but I would like to call the attention that we can only do type of research like that and to answer questions like that because we are not alone. We are not doing this only with the data in Brazil, but it's already a collaboration through IRC5. So to be able to really have more subjects with actually protocols that were done in learning, and then we can combine the data to be able to answer questions that are more important to understand the impact, for example, of imaging or structure or functions that we see by neuroimage into behavior as well. So this was just presented last week in the Human Brain Mapping Conference, the Organization for the Human Brain Mapping, and actually I'm very happy to be able to present the data that was put together now. So to finish, I will just, Lynn asked me to talk about these possibilities to explore and to understand how MRI can be used as a tool to understand our brain functioning and structure. And as I have a couple of finished? Okay, so I have a couple of more minutes. I just want to show something that's we never, and I think this was never done in patients with callosogenesis, but it shows how we can actually modulate and map the chains of our brain in real time. Here we have an MRI, and with a very rapid sequence that we do very, very, very fast, and with a computer that can process the signal, and can put it back to the subject that's inside the MRI, we can do what we call a neurofeedback fMRI. We can show the activation of a specific region or a couple of regions of your brain in real time and give you, for example, information like a thermometer like that. Okay? So here, and then, for example, let's see. If we are ask the subject just to imagine that he is moving the hand, doing these movements, and then you have an expected activation related to the region that is controlling the movements of your hand. And we can show how much is this activation, and because you have this feedback, you can improve the control over your brain. And there are a lot of people working with that. And actually, what we and other groups also now already shown is that we can do that, and we can train our brain in sessions like in half an hour. Of course, it's a very simple training. This is only motor training and very simple. But we can show with MRI is that you change your brain connectivity, and you can also change your brain connectivity. So those tools are, of course, only for research, and can help us nowadays to understand how we can modulate, how we can improve the modulation, which regions we would like to modulate or we should go. But again, it's a future that's coming, and it's just possible because researchers from different areas, engineering with clinicians that can actually work on that. So this is a happy way to show that our brain is changing, can change, and we can map those changes in real time and inform ourselves about those changes in the brain. So I would like to thank a lot IRC5, NODCC, and a special thank you also for the DCC, that's the organization in Brazil that started a couple of years ago. So it's very young compared to the NODCC and also from NODCC, but they are very important, And I would like to thank all the help and the support that they have been actually done in Brazil to be able to do awareness of that, and to also be able to join for efforts with actually now nice equal to US. Brazil is a very continental also country, and we have very diversity everywhere. So access is very difficult in a couple of states in Brazil. So I'd like really to thank DCC, and those are just a couple of pictures from last year event. I'm not sure how many of you guys were there. We would love to be able to host in person sometime. So it was done in Rio, but not really in Rio, as you can see from the pictures. But on the other hand, we had a lot of partners and collaborations for around the world, because this was Zoom. And nowadays we are here in a hybrid conference, but I really hope that you can visit Brazil, and I would love to host it again. Thank you so much. We have some questions from the Zoom. Yeah. First, I just a comment. I can see myself on last year's conference picture. That's great. Thank you. I hope you enjoyed it. Very good. So we have a question here. Can you see the level of myelin on MRI? Was there research on the role of myelin in developing neuroplasticity with ACC? So this is a very, very good question. So all the things that we are measuring with the diffusion is related to myelin, but not only to myelin. We have those images they are produced, taking into consideration the white matter tracks. So we have the real axon, the real neuron part, but also myelination talks is also very important for those type of measurements. However, there are a couple of studies and a couple of processing that can actually help us to guess if it comes really from myelin or not from myelin. So there are a couple of ways to really answer those questions. It's not so straightforward though. And there are not only diffusion, but other types of sequences like with magnetization transfer and other type of sequences that can also measure myelin. So yeah, you can, but it's not that straightforward. Yeah, this was a question. Yeah, and also, and if you have myelin in developing neuroplasticity in ACC, I think not that I'm aware a specific work on that. The other one is I'll be moving to just
I think it's really exciting, all the research that you're doing, and you were talking about the induced brain plasticity, and I've heard of other researchers like Elon Musk is doing something kind of similar to that, and I was just wondering if that does grow, where would your dream be that it would take you to, if you're putting things on people's brains to help them create muscle memory? It's just very exciting, and I think people should pay attention to that if they've got children with, there's no magic, but it's really exciting to hear these findings.
Yeah, thank you so much for the question. Well, I think that studying and understand the brain plasticity is a way to be able to do and to dream for therapeutics and to not only intervention. There are a lot of ways to challenge our brains. Yeah. So my dream is, of course, that we can help people, but I think that's a long way to go. I think that we are still trying to understand how it's happening and how it happens. And of course, it might be that also with the advance of the research, we can also take to a personalized, for example, more therapeutic for each one. So this also can help us to understand if this is a real good therapy for somebody or not. So mapping this as fast as possible can guide us and can actually make us to help more with specific treatment, specific person, for example. Perfect. Yeah. So Lynn, I'll just repeat. Lynn's just mentioned what are the value of those type of images in the individual level. I think that's very important. Almost the majority of the things that I showed here, especially with the brain maps, they are done with group of people with one condition compared to group of people with other condition. Doesn't mean that we don't get interesting and important information from there. We are understanding how, because those types of things, the differences are very little sometimes that we need group of people compared to group of people with a different condition. So we get important information. But we are getting important information from the group level and not to the individual levels. Of course, when we see a Probes bundle or a sigmoid bundle in one specific subject, we are seeing in this individual level. This is true. But the majority of this group of analysis with these advanced neuroimages, we are talking to group level and not individual level. Yeah, I think I...
So can you tell from a brain image how my brain works?
Elise is asking if we can tell from a brain imaging how the brain is working. This is a very, I will try to respond in two ways. If I'm mapping, if the question is, can you map where is the area that controls my hand movement? Yes, I would say yes, I can map where is controlling your hand movement. Can I map where is my cortical visual cortex, where I can actually see? Yes, we can do that. So functional image can do that type of information. Sometimes it's very useful. You can imagine if somebody will do a surgery and will extract, for example, a lesion or tumor. It's important to know if the tumor is very, very close to the area that controls my movement or not. So you can get information from the single level. But for complex functions, then I would say no. Then again, in this group of individuals. So if you are going to go to more complex questions, how is the moral judgment? How you process some information in a much more complex way? Even sometimes language, you need to have group of people with this condition compared to group of people with other condition. And then you can get good information, but you need to interpret that with cautions and with all the limitations of that. But you can get, oh yeah, you can get that from out. Oh, yeah.
With MRI, so yeah. We can record activity. Activity is much faster than functional analysis. Actually, real action potential, they call it, as the brain activity transmits throughout. That is much faster than visualized in MRI.
Yeah.
Even what you know, but not at the kind of resolution and by putting an electron.
Yes. Yeah. What I can repeat here in the microphone so people on Zoom can also listen. So what Linda is saying is completely true. Like we saw the reconstruction, for example, of the bundle with histology, and then we go to a different level, you can now actually also in animal models, you can record the activity even from one single neuron and to understand the activation at that level. We have two limitations in terms of acquisitions of functional imaging with MRI. The resolution and also the timing, as you said. So it's getting faster and faster and faster, so that you can do like the real-time processing. It's much, much more advanced than it was before, but still, it's not even a little bit compared to a recorded in an animal model, for example. Any other question? Okay. Oh.
What about the speech language part that the brain can use now?
Yeah, very, very good. So actually, Lynn also asked that. So the majority of the mapping, also of the language, is also in the real process of the language is also done at group level. So group of people, typical people compared to group of people not with callosogenesis, for example. Dr. Scherer has actually one study exactly asking that type of questions. For lateralization of language, in a couple of also pre-surgery programmation, for example, you can also understand like just what are the level, what brains, what hemisphere are controlling. But then it's very, you are not really understanding the complexity of the processing of the language itself. Yes.
The reason I bring it up is because often when people come to do research with us, they'll ask, can I see a picture of how my child's brain works? Because we show these really beautiful, fancy pictures, and I want to set a realistic expectation about what we can glean from an individual's images. We get information, important information, but there's also things like that area of the brain that's handling language may be in the same place as everybody else's, and it's working really hard, but the level that it's working at doesn't necessarily associate with how well it's functioning and how well the person's reading. So those are the kind of questions that are complicated. It's not, oh, if there's action there, then more action means better action, better functioning. Does that make sense? So it's really complicated to break it down at that